Hello everyone, my name is Philipp Reschneider. I'm a bioinformatician in the genomic applications team here at Oxford Nanopore, and I want to welcome you to today's masterclass on how to perform tumour-normal sequencing. So, as you may know, if you've seen earlier masterclasses, for what we want to do with the masterclasses is we want to show you the full workflow, basically from sample to answer, and that includes preparing your library, sequencing it, and then analyzing it. One thing that's special about this masterclass is that it's focused around the worked example, and that's about tumour-normal sequencing. So let's start at the beginning. What do I want to tell you today? What do I want to show you in this masterclass today? So first of all, I want to walk you through the benefits of nanopore sequencing for cancer genomics. And then, as I said, I want to walk you through the whole workflow from start till end on how you do tumour-normal sequencing with Nanopore. And in the end, I want to use our worked example, our example dataset, to give you a glimpse of an insight into what kind of results you can get if you were to perform tumour-normal sequencing with nanopore sequencing. But so let's just dive right in. And let's start with why Nanopore for cancer genomics. Well, so Nanopore comes with quite a few kind of like unique features. And one of them is that unlike other platforms, the read length is not determined by the platform, but actually by the library. So you can sequence very short reads as well as very, very long reads. And also you can sequence DNA and RNA directly, so native DNA and RNA, which means you can skip PCR, there's no biases, and you maintain your DNA modifications, which is especially important for cancer. And then last but not least, the platform is very flexible. It's very scalable from kind of like portable to really high throughput. So how do these transform into kind of like cancer genomics? Why are they important? So from my point of view, what I find most exciting about using nanopore sequencing for cancer genomics is that a single platform with an instrument that can basically sit on your desk can give you an insight into a wide variety of different things. And so, for example, you can get smaller SNPs and indels, but you will also have access to large structure variation or genomic rearrangements, which are very important in cancer and are known to be very important. But not only that, on top of that, you can use the long reads to phase those variants and to understand better kind of like the haplotype structure and the cancer progression and how these things change during cancer progression. But that's really only the kind of genomic changes. On top of that, you can also get epigenomic changes, as I said before. For example, 5MC and 5HMC, and they're also known to play a key role in cancer. And I'll give you an example on that later. And this kind of basically comes for free on top of all your sequencing. And then, in addition to that, something we're not going to talk about today, but there's obviously the whole world of RNA or cDNA sequencing. And with long reads there, you can look at splice variants, fusion genes, and then in addition, in the kind of epigenomic space again, with our POR-C protocol, you can even look at chromatin conformation. Okay, so loads of possibilities there, but let's start kind of like more conceptually on what is tumour-normal sequencing before I go into more details here. So as the name says, we obviously sequence like a normal sample. And the normal sample is basically just a healthy tissue of the affected person. And if we sequence that and look at that, what things that we would expect, for example, are variants, obviously, compared to the reference. Here we have two SNPs, one homozygous in both haplotypes, one heterozygous. Again, completely as expected. These are what we call the germline mutations, the germline variants. And then also one thing to keep in mind is that in a normal sample, we expect roughly the same number of reads from both haplotypes. So roughly 50-50. So this is, if you will, our baseline of the mutations or kind of like the genomics and epigenomic imprint of that person we're looking at. And then, obviously, we'll also sequence the tumor sample. And so when we look at the tumor sample, one thing that kind of stands out immediately is that the germline mutations are there as well. It's to be expected because those tumor samples are derived from healthy samples from cells from this individual. But on top of these, we also can have a wide variety of other so-called somatic variants. So here I just show kind of like a depiction of a structure variation, a SNP, and modifications. So as you can see, they can also, since it's cancer, they don't have to be in all of the reads in haplotype, but they can be low allele frequency. So that's one of the things about somatic variation. So again, here we look at SNV, indels, SVs, and modifications. But one other thing to keep in mind when looking at this, so cancer is known to have like large-scale genome rearrangements. So like one haplotype can be amplified, and then you'll get more reads from that. And this is what you can see here in this example as well, where 70% of these reads in this region come from one haplotype, whereas only 30 come from the other. So this is roughly what you can expect in cancer, and this is what we're going to look for the rest of this presentation. Right, if you've seen a masterclass before, one thing that we typically try to do, or kind of like encourage you to do, is just start at the end. Think about what your question is, what are the things you want to answer, and then work backwards to kind of inform the bioinformatics that you need, inform the amount of data or read length that you need, inform the library prep that you need, and so on and so on. And so this is what I want to walk you through now for a tumour-normal example. So if we think about this for a tumour-normal experiment, what are the main considerations? So what are the experimental goals? Basically what I just told you, we want to find the somatic variants in the cancer. We can do germline as well, and they can be informative, but our main focus is on somatic here. What are the regions that we want to look at? Actually, for this, we want to look at the whole genome to understand basically in as much detail as possible what's going on in the cancer sample. And then obviously, as I said many times now, we have tumor and normal samples. So this leads us to how do our samples look? Obviously, there will be human tissue samples, it will be clinical research samples. So with this come a few things. For example, in terms of DNA, we can have variable molecular weight, and we might be limited in terms of sample amount in some instances. So these are things to keep in mind. And then last but not least, kind of like more practical considerations, how much sequencing, how much depth do we need? We want high depth on these samples, and we don't have to be doing this out in the field. So kind of these are two other considerations to keep in mind. So this is again like the end, roughly thinking about what we want. So next, kind of like, how do we get there? How does this translate into experimental workflow? So we'll start with DNA extraction. The main goal here is you want to extract high molecular weight DNA. And so if you look at tumour-normal, obviously as I said before, you will have different tissue. The tissue you have, it depends on kind of like what tissue the cancer affected and what kind of cancer you want to study. So it could be a whole range of tissues. And luckily on our community, on our Nanopore webpage, we have recommended extraction protocols from multiple different tissues. So you can go there and look up the right one for you. You'll find lots of other guides in terms of handling and storing DNA, which can be valuable as well. So as soon as you're finished with extracting your DNA, the next step is QC, and that's an important step. There are two things we generally recommend. First of all, running a qubit to assess the quantity of DNA to make sure that the right amount of DNA goes into your library preparation. And second, run a nanodrop to assess the purity of your sample, just to make sure that nothing goes into your library prep that you could reduce efficiency there. The second consideration is around read length or fragment length, and that comes with one of the great characteristics of nanopore sequencing, which I've mentioned before, meaning that the platform doesn't restrict the read length, but it's determined by the sample itself. And there are a few things that you can do, and two I have here. One of them is light shearing, and the other one is size selection. Light shearing we have found to generally improve output, and size selection obviously helps by removing shorter fragments in your sample. On the right, you just see an example of basically the same sample prepared in different ways, yielding different read length distributions, just showing the amazing flexibility that you can get from the Nanopore platform to tailor your sample or your read length distribution to your application and what you need. For our example here, which is tumour-normal sequencing, we generally recommend roughly like a read and 50 of 20 kb as a good starting point. It's not kind of like a hard restriction, so even if you have more fragmented libraries with shorter read length, you can get tons of information out of your tumour-normal sequencing, and I'll show you an example of this later, but 20 kb is a great starting point. Overall, our recommendation would be light shearing and size selection. But again, it really comes down to what you get from QC. You need to look at your sample and make these decisions. And I'll show you an example of us doing that later in the worked example. As soon as you have your DNA extracted and fragmented, we move on to library preparation. For our example here, it's quite simple. Our clear recommendation is the ligation sequencing kit. It's a good balance between read length and sequencing output. Just as a reminder, it's a roughly 60 minutes prep. It's PCR free, so you don't have any biases, and you maintain the modifications, and you have complete control or good control over your fragment length distribution, so it perfectly fits the bill here. There's not much more to say about it really in this case. The only thing is that if you have your prepared library, obviously you can move on to sequencing. And so, as I said before, one of the strengths of Nanopore is that it's an extremely kind of like flexible and scalable platform. So this here shows you the range of devices that we have. And it starts on the left with our portable sequencers that are based on our MinION or Flongo flow cells, and moves to the right up in terms of throughput to our high throughput platforms, the PromethIONs, which are all based or using the PromethION flow cells. And the cool thing is you can kind of exploit this scalability for tumour-normal sequencing in a couple of ways that I want to show you in the rest of this presentation. The first one is actually kind of like in-depth QC. So I've told you about kind of like tailoring your fragment length and all these kind of things. One cool thing that you can do as soon as you have your library, you can take a small amount of it, you can put it on a Flongo, and you can actually sequence it. And if you do that, this will give you a good indication of whether your read length distribution and output is actually roughly where it should be given your actual libraries. So this is optional, but it's still a pretty cool thing of doing in-depth QC for your samples. As soon as that's done, obviously I've said we want to have high-depth data, so we'll be moving on to our PromethION platform for tumour-normal sequencing. When it comes to selecting the right PromethION device for you, it really comes down to the scale of your experiment. Basically, how many samples do you want to run in what time? As you can see, we have the P2, which can run 2 flow cells at the same time, and then the P24 and the P48, which can of course run 24 and 48 flow cells at the same time. All 3 PromethIONs will use the same PromethION flow cell, so for all of them you will get the same high output per flow cell. But obviously, if you want to run a low number of samples, the P2, as you can see on the slide, will enable you to roughly run 200 flow cells a year. But then with the P24 and the P48, you can scale this up to roughly 2,500 or even almost 5,000 flow cells per year, allowing you to really tackle large numbers of samples. Then one other consideration here is that generally for tumour-normal sequencing, we would recommend using our flow cell wash kit, which allows you to maximize the output you get from a flow cell. And then if you happen to have very precious sample or very limited sample amount, you could even look at our library transfer protocol, where you can retrieve the library after sequencing and move it to another flow cell. Let's say sequencing is done, we chose our PromethION, we ran our samples, we got the data. What's next? Well, obviously data analysis. So by now there's a wide range of tools available for tumour-normal analysis and somatic variant calling tumour-normal samples. I've put here a couple of tools that we have used for our analysis just as a starting point, and a few kind of like honorable mentions, if you will, on the side. So in terms of SNPs and indel calling, our clear recommendation is ClearS. And then for structurization, we're currently using NanomonSV. Savannah is a very good tool as well, worth a look, but we're currently using NanomonSV. So again, these are for calling like SNPs, small indels, and structural variation. WhatsApp is a very useful tool for phasing, so you can phase variants and reads. So you could do things like you can phase the tumor reads based on the normal SNPs, which is something that you often want to do. And then you can actually go on and use these phased reads, even to call kind of modifications on these reads in a haplotype-resolved fashion. And for this, we recommend ModKit, which will replace or is replacing our current tool, which is ModBAM2BAT, and then DSS to get differentially methylated regions, basically to find interesting differences between different tumors or tumor and normal. And then kind of like in the bottom, we have kind of an in-house CNV calling pipeline that I will talk a bit more later. That is very useful. And then if you want to look at more specific things like ecDNA fly, it could be a good option for you. So as I said, there's a whole range of things that you can do. One thing to mention is that we're currently wrapping this tool up in one unified tumour-normal somatic variation workflow to make it easier to run these. And if you watch this kind of at a later time point on demand, please feel free to go to our Epitome webpage and to see what we have on offer there already. Right, so one of the things that one of my teams is doing at Nanopore is actually like looking at these tools and testing that, and finding out kind of what performance you can get out of these tools, just as kind of like information for you, so you know what you would be getting. And so just kind of like quickly spend one slide on this. We typically call it benchmarking. So this is around benchmarking of somatic variant calling. So here on the left, we have SNVs, so small SNPs and indels. And what we did there is we took two well-characterized healthy cell lines, where we know what variant should be there, and we mixed them in different ratios to simulate different allele frequencies. Then we ran CLARS on these, and we compared what we got to the truth set. And just kind of like as a quick indication, we did it at different depth. And one of the takeaways is that you get, independent of allele frequency, you get really high precision. And down to 20%, you also get really high recall and F1 score with that. On the other side, we've also started testing benchmarking somatic SV calling. Somatic SV calling is generally kind of like benchmarking somatic SV calling is a bit more difficult than SNVs, because we can't really do the mixing of healthy cell lines, because tumor cell lines just have so many more variants that or structural variants that you wouldn't see in a healthy sample. So we rely on a truth set that exists, kind of a multi-platform truth set, and basically chose high confident variants, split them by allele frequency, and what we found is that down to 10% of allele frequency, we found more than 98% of the expected SVs. So in our case, we basically just missed one here. So again, this is just kind of an indication of what performance you would get. And this also concludes kind of like the first part of this talk, where I want to show you, as I said, kind of like the full workflow, kind of like what are the options that you have. And then with the next few slides, I want to kind of like take all of this and transform it into one worked example, walk you through the considerations that we had for those samples and the decisions that we made. So again, let's begin with the question of kind of like what are the answers that we want to get out, and how does that influence our considerations for the experiment. So one thing I need to mention here is that we did not have direct access to clinical research samples for tumour-normal. So we had to fall back to commercially available DNA. So for these samples, unfortunately, we couldn't do the DNA extraction ourselves. What we did was the QC that I talked about. And what we found is that for these samples, the fragments were relatively short. They were a bit more fragmented than we would have wanted them to be. So basically what we did, we found that there is no need for light shearing, because we wanted to maintain the maximum read length of those samples. And then also the quantity or the sample amount we had was very limited. So in this case, we decided to skip size selection. Again, this is just an example to show you that while our standard recommendation is size selection plus light shearing for tumour-normal, it really depends on your sample. So you need to look at the QC and then make decisions based on that. And this is what we did for our samples here. Then moving on to the library prep, again, the decision there is pretty easy. The ligation sequencing kit is the clear choice here, and this is what we did. As a reminder, it's a good balance between read length and output, and usability and ease of use. Moving on to the sequencing, as I've kind of indicated before, we want the PromethION platform to do this experiment. On the next slide, I'll show you how many samples we had, and just based on the sample number and the turnaround time that we needed, the P4024 was the best option there. And then last but not least, data analysis. Obviously, we used the kind of like somatic variation toolkit that I've just showed you before on the slides. So we used the tools that I showed you to call basically as many types of variants we could on these samples. SNPs, indels, SVs, 5MC, 5HMC, CNVs, everything, and then kind of like had a look at what we could find in these samples. Right, I keep talking about the samples, what did we actually look at? What did we have? So very early on, we knew we wanted to have multiple tissues because there are very interesting changes, especially in modifications between different tissue types. And then to make that kind of like get more meaning or almost statistical power out of it, we wanted to add replicates. So we basically choose different tissues and 3 replicates per tissue. So these are kind of like not replicates of sequencing the same sample, but it was 3 different tumour-normal pairs, but from the same tissue. And the tissues that we chose were kidney, lung, colon, and breast. For these, we had normal and tumor samples. And then in addition, we also decided to add some brain tissue samples. We couldn't get brain tumor samples, but brain has very interesting modification patterns, especially in 5-HMC. So we wanted to still have them in, even although we didn't have tumor samples for those. In terms of read depth and read length, especially in terms of read length, you can see that we had an N50 between 12 and 15 kb. So it's a bit below of what we would expect, the 20 kb that I mentioned. But again, there was a limitation imposed by us having to go for commercially available DNA that we couldn't extract ourselves. But still, nevertheless, as I'll be able to show you, even with this read length distribution, we were able to get tons of interesting data out of these samples. Here's just kind of like an overview of read length and depth per sample. As you can see, the read length changes a bit. So these, again, it depends on the fragmentation of these samples, and these were quite different samples and tissues in a sense, so this makes sense. And then in terms of read depth, what you can see is that we aimed for kind of like roughly 30 to 40x for the normal ones, and then for the tumor ones, we had some that were higher, like above 60x, some at 50, and then some at kind of like the same range of the normal samples. Basically, this gives us a kind of like relatively broad range to look at the effects of different coverages as well, which was very nice. Generally, kind of like having a bit more sequencing on the tumor makes sense because again, that makes detecting those somatic variants easier. Right, so this is kind of where we started. This is the samples that we have. Obviously, as I said, this is kind of like a treasure trove of interesting things to look at and discover. Unfortunately, I won't have the time to go through all of them, so I picked a few kind of like representative examples. And so what I want to start with is large-scale copy number variation. So the reason for that is that kind of like genomic instability is a really well-known characteristic of cancer samples. So in cancers, it's super common to have copy number gains, so you kind of like whole chromosome duplication or chromosome arms or megabase regions that are duplicated or lost. And so they're interesting because these large-scale changes obviously can affect, for example, the dosage of multiple genes at the same time, or they can get rid of multiple genes at the same time if you just lose the whole part of the chromosome. So from that perspective, they're very impactful, of course. And to study this, what people often do is just do low-depth sequencing, so roughly 1x on the cancer. Because even at 1x, you can get fairly good copy number profiles, not in a phased or haplotype-specific way, but just in a haplotype-collapsed way, as you can see here. And so I've told you can kind of like exploit the scalability of the platform in multiple ways. This is another way that you can do. Since you only need 1x, you can easily do that on a MinION, and you can potentially do it for multiple samples on a MinION. And where I've shown you kind of like QC for your library on a fungal, this is really QC for your tumor sample. Like for one thing that you can get here is tumor purity estimation as well. So for this sample, it was at 72%, meaning in your tumor sample, 72% of the DNA roughly is from the tumor, and the rest is from like normal cells that are just surrounding there. It's very common. And so this is just a way where you can quickly check if the sample you want to sequence has high enough tumor purity for you to be informative, basically. What we can see here, just kind of this is technical view. The more sample or result specific view is that for this sample, we know from the literature, this is a kidney cancer sample, that kind of gains of chromosomes like 6 or 12, oh actually no, it's 7 and 12 and 16 are well known to occur in these cancers. So what we see here makes a lot of sense. The one thing that did catch my eye was this small bit here. It's a bit uncommon because there are two things that can happen. So basically, we don't know at this point whether the two larger bits on the left and the right were just duplicated separately, and the one in the middle just stayed the same. So you have one copy from both haplotypes and it just stays the same. or if both haplotypes were amplified, but then one haplotype was lost, so you have 2 copies of one and 0 copies of the other. And so why is this interesting? So basically that leads me to another common trait of cancer, which is loss of heterozygosity. And so that can kind of impact allelic variation in a lot of genes, and therefore is an interesting thing to study. But just from what I've shown before, we can't really tell if that happened here or not. But if we go to a PromethION platform sequence at higher depth, we can get a much clearer picture, a haplotype-resolved picture. So that's also why I put the PromethION here again. And what we get here is, so this is the CNV pipeline that I mentioned earlier. And what this pipeline can do, it can give you clone and haplotype-specific copy number variation. Here we're just looking at haplotype-specific calling, because there was only one clone in this cancer. Again, tumor purity matches what we've seen before. And then you have 2 of these profiles, one for haplotype 1 and one for haplotype 2. And as you can see on the top in haplotype 1, in chromosome number 7, the full chromosome was duplicated, so we have 2 copies of that haplotype. On haplotype number 2, like, the chromosome was duplicated as well, so you have 2 copies, but this small bit was lost. So in this small bit, we don't have anything from haplotype number 2, so we have confirmed loss of heterozygosity in this region, which is interesting to know to understand what happens in this cancer. So this is all really nice, but we're with long reads that map uniquely across even repetitive regions. So the question is, can we detect the exact breakpoints for this event as well? So that's what we want to look at next. And so for this, we zoomed in, and what you can see here is that on the top you have the normal sample, and blue and reddish are the 2 haplotypes. There is no unusual signals. However, if you look at the bottom to the tumor sample, you have this clear breakpoint in one of the haplotypes. And this is one of the breakpoints of the deletion that caused this piece to be missing. So we also found that the other one is in the centromere, which is very common in these cases. And we also confirmed that with SNP frequencies. So from all perspectives, that does make sense. And it's interesting to see, because this means there are, for example, 4 copies of the MET oncogene, only 2 copies, but with loss of heterozygosity for the EGFR gene. So these are things that you can then kind of continue to work with, try to find out what functional effect that would have. For me, in the instant of time, I need to kind of stop talking about genomic changes. We can obviously do SNV calling and somatic SV calling here as well, but I don't have more time. So I'll move over to kind of modification changes. And so why am I doing this? So first of all, this is kind of to me, some kind of game changer really in terms of Nanopore. If you run a Nanopore cancer data set, you get the modifications. If you don't, if you just do it the way we just told you. And so just as an introduction, there are 28 million CpGs in the human genome, and 70% of them are methylated usually. This is what you see on the right here, just kind of a 30 megabase region of modification levels in the different tissues. You see they're relatively similar, high throughout, which is to be expected. Those regions are often interrupted by short unmethylated bits of CpG, and these can be kind of a binding platform for transcription factors, so these are very important. If you look at cancer for comparison, you see that overall the methylation level drops, and that's known, but this is often kind of interrupted by regional hypermethylation of promoter regions, and these can have an important kind of functional effect. And so this is one of the things we're going to look at next. This is one example in colorectal cancer. And if you look at this, it's visually very obvious, but let me still put the box around it. What you can see is that in these 3 samples in this region, there is a high methylation signal, whereas in the others, there's none. And interestingly, this is only the case for 3 colorectal tumor samples. And if we look it up, this promoter region for this gene plays a key role in colorectal cancer. It even is connected to 5-year survival. So this is a very key and important finding. And then the other thing here is that this 5MC signal is really strong. But if we look at the CNV signals of these same samples, they're actually not that clear. There's not so much in the CNV profile there. So this, to me, just kind of enforces the importance of looking at both things, epigenetic and genetic changes at the same time. So this concludes kind of the examples from our work example. There's much more I would have, but in the interest of time, I really have to move on. And so this was a lot, obviously, I went through, but luckily we have lots of resources on our webpage for you to look at. So if you're interested in cancer research, there's a cancer research page. For protocols, as I said, we have a lot of protocols available in our community. We have loads of workflows and guides and white papers around different topics on the web page, and a special section of the community about the analysis. So please go and check that out. So this kind of brings me to the end of this masterclass, and I hope what I was able to show you is, first of all, kind of like what a comprehensive look the Nanopore sequencing platform gives you on tumour-normal sequencing and cancer genomics in general, and then show you basically end-to-end how you could do your tumour-normal sequencing yourself with Nanopore, and then give you a glimpse of the results that you could get. For example, low-depth CNV calling as a kind of like sample QC, kind of like as I mentioned before, but then much more kind of like in-depth look at haplotype-resolved, copy number variation changes, and also modification scores. So with this, thank you very much for your attention.